everyone. In the interest of time, and not to penalize those who were in time, let's get started. It's my great pleasure to have a friend and colleague and partner in many crimes here, which is Roland Grafström. Roland is as close to the Nobel Prizes as can be. He's at Karolinska. Whether his work is close to a Nobel Prize, you have to judge yourself. But actually, he has a history with us. Shortly after his postdoc at the NCI, the National Cancer Institute, he was here the first time talking in 1980. So this was 1 year before our Center for Alternative to Animal Testing was created at Hopkins. And so you can imagine this is for me incredibly long. I was still in high school at that time. Okay, and he was already a postdoc. Good. Roland has been doing very similar things. We have been interested in integrating omics technologies, data analysis, and making sense of all of these. And you will hear this, so I'm very happy he's here. I will not take any more time from him. He is moving in between Finland and Stockholm all the time. He has a lot of interests. Among them, fun fact, he's owning 2 Porsche race cars, which I had the privilege to see. So he is a speedy guy, and you will see that he's doing also a lot of research with fast velocity. Okay, Roland, the stage is yours.
Thanks very much. Thomas was not supposed to talk about the cars, but anyway. So I'm very glad to be here, and obviously you heard already that it's a history of when I was doing my postdoc, and I also have a pleasure of having colleagues and other people who were postdocing at the same time visiting and right now. So it's a great moment, and great to see all of you here as well and take the time to hear this. I'll talk about tiered approach assessment, and hazard, of course, is to understand the inherent toxicological properties of whatever you're studying. It's drugs, chemicals, but in this case, materials. And we do have an enormous need of high throughput of information and data. And as Thomas says, you're very much aligned with that idea. And of course, then we also need to use the modern technology of interpretation, the AI, etc. So you can see the title here is by high throughput screening and omics and combining that. And what you're seeing is what I used to say at younger age, my co-workers, but now my collaborators, because I'm a senior scientist, as you understand. And the work that I'll talk about primarily is where Vesa has been doing and leading the high throughput work, Vesa Hongisto, and Pekka, who is the expert in the omics side and the informatics and AI side. And just before I go on, you have this term conflict of interest. And when in my situation, I think it's nice that you know that we have an innovation office and all the Swedish scientists own their own inventions. So we have a very totally different system for the scientists. They can explore it, and it's positive to do also sites, tools, and consider commercial aspects of it. So by that you understand I do have what you term a conflict of interest. Also, you see that I'm involved with companies, including one company who is made to explore omics data that we have in Sweden. So let me go on with that. I guess that's right here. No, I don't change the slides in this one. The slides here, maybe. No? How do I change slides? Like this way? Not that way. It usually is one way or the other. So,
We also have a problem with the screen is split, so apparently.
I don't. But this one is it? But the pointer worked. So I see the pointer, and then maybe okay. So how do I just do it there?
Spacebar.
This one.
This one here.
This one. Okay, really good. All right. So I will share with you our concepts explored, of course, here as well. And maybe that helps also for the understanding. So high throughput systems toxicology aiming for animal-free risk assessment. Risk assessment then is a bigger process, of course. You really want to be able to say that if someone asks you, is this substance, is this material toxic, or can I use it? It can, and saying that something is safe is actually really very complicated. So with many processes that I touched upon. I'll share you first now the sort of biology here, which where I sort of stand for with my team and collaborators. And this is doing the high throughput concept, which is here. There's a lot of information here, but really don't mind about that. So multiple, in this case, materials, multiple concentration, using many cultures simultaneously, studying that, getting a readout and interpreting that from the dose response curve is a very important early thing. And you move away from traditional cell culture where you get only a few data points, in this case with the high throughput idea, to high throughput screening, and you get many thousands of data points. I'll share also with you as I go along a little bit on the side of that of data points. How many are generated. So that is the first tier. It's a sort of idea of what is also called an IATA, an intelligent approach to testing and assessment. And then you move on to those which you really believe are to be the most, where you have worries. You don't know really what they do, and they come out from this first evaluation as being very active, maybe very toxic, or maybe have particular properties that you are afraid of from the health perspective or the environment perspective. And then you go for those, and you of course know a lot about the idea of from the first round, and then you go much more deeper with the omics technology. Hopefully you have heard about omics technologies, all of you. And I will share with you a tool that we developed called predictive toxicity tool, very much coming from the ideas of that we can have a toxome and things like that, which you also, you at this department here have worked on many years ago, and then how that is used and so forth. So the majority of the presentation will be on this from a project called HARMLESS, which I think is quite cool and ongoing right now. And then I'll share with you how you can move all the way from the omics, interpreting that on something called AUP, the Adverse Outcome Pathways Framework, which I'll explain also a little bit. And the idea again, you now with the new technologies, you go much faster. And you can get a lot more data. And then, of course, you need to have informatics and tool to interpret all of that data. Advanced materials is in the title. So what is that? We have very much an increased societal use of that. And there is a working definition. Most of it comes from the nano, that materials, when they come to a very tiny size, have different properties. And then, of course, the idea is to use them. So it emanates from the idea of being very small can make you do things that you can't do at a bigger size. So it's understood as materials are rational designed to have new or enhanced properties and or targeted or enhanced structural features with the objective to achieve specific or improved functional performance. So we're getting into also here safe by design. And I thought, thinking of being invited here, that materials for this engineering side of your department, that maybe this also could be kind of interesting to hear about. So safe by design concept is there. And one part of that is defining non or low toxic or inert materials, because then we are much more safe, of course, to use them. But it's a lot more than that. And that comes in a slide or so. And materials, if you analyze the toxicity, which then is sort of my specialty, then it's a lot about size and shape, surface chemistry, solubility, ion release. Then there's also biologic interaction. Macrophage-like cells, for example, they really respond to materials like it could be bacteria, so they take them up. So macrophage-like cells in the lungs when we inhale materials are maybe more target or can give you more information. So if you study these kind of things, you like many different cell models. that represent different part of the lung, for example, or just have different properties dependent if they're sort of normal, immortalized, or tumor-like, etc. And then you have many aspects of materials, which also makes it very tough to do. The aggregation, how do you deal with them? What kind of protocols do you do? And in the end, as with everything you study, whether it's drugs, chemicals, or materials, you have the aspects of how clean are they? Do I really know what I study? And these kind of things. And here's a recent article, which I'll have also on the next slide, which gives you a very current update on these kind of things. All right, the bigger framework is right here. Then if we want to study materials, advanced materials, this is the abbreviation. And here we talk about next generation risk assessment. It's sort of a popular term. But what is next? Well, it's next. Who comes first? I mean, it's sort of a popular term to seem to be cool to everyone, I might. But anyway, so you have a problem formulation first. You have an idea, where do you come from in the safe by design? What are the consideration of exposure, synthesis, making it used, and life cycle? Life cycle is an enormous thing to study, very complicated. I'm glad I'm not the expert on that side. I'm an expert on something else. So anyway, from the overall, you consider green or red. Do I move forward when you have a material and you want to use it? And then you have this whole process, and it's probably very logic, right? What do you know? What do we know? Data gaps? What is there already? Can we use this material which we are believing to be very good in a context? And then we, of course, have gaps most of the time with something new. And then we have to look into the acquisition of new data. And if you see very carefully on this big slide, all of what is below on that slide and in this area is where I also work. I will show you results. Then you have to evaluate all of that, and you have to make the assessment, and then sort of, do you go on or not? And what I particularly like here, that was developed actually in a project called Gracious that I was not involved in, was this simple terminology when you work forward. And that is when you do this I8 then testing assessment, and you combine NUMs. And NUMs is new approach methodologies. How many of you know the term NUM? Can we get a hand up? Okay, some of you have a new term now that you need to know. I've had an earlier talk this morning with someone very nice. I said, take with you that term, NAM, NUM, new approach methodology. And it really is in vitro and in silico models, and in combination of cells and biology and computational and AI and all of that, for those of you who are not sort of familiar with it. So anyway, what are the cool thing here? What they are, where they go from the environment point of view, where they go from the biology point of view, and what they do. And see, this is where I come in. What they do primarily. This is the work that we just had, the slide, that tiered approach that I had in the first really large slide, getting that first idea, evaluating lots of materials, and moving to a lower number to move forward. And then, of course, there's assessment of all of this, but this gives you sort of hints. So I'm a little bit slower now. I'll speed up a little bit, but I want to keep most of you in a little bit of longer time than just a few slides. I'll do my best. Okay, so the HARMLESS project, this is the one that I think is where it's really in its end, actually. It's in the 4th year now, and we've generated lots of data. And from many projects where we are involved, I felt this could be a good example to share with you the type of work that is ongoing on the European side in big, larger projects. So this is something like $8 million comparative budget. And I'm responsible for the data generation in this project. And here are the aspects where it comes to these high throughput and modeling key issues. So we're using these new approach methodologies, doing hazard estimates, the inherent property of sort of danger and and toxicity for about 70 materials. And we have a particular scoring where we look at certain things, and we do begin with that for dose response assessment. I'll show you this in a couple of minutes. We apply several different cell line models. One is immortal, one is two are tumor, one is from liver, one is from lung, and we have a macrophage-like line. You can see what I mentioned, macrophages before. And what we do, we really work up and down the point of departure, the dose response curve, and see where do we have an effect in terms of dose, no effect, lowest observable effect, no observable effect, etc. Show that in just a moment for you. And that is, of course, to define the causative concentration, the potency, and understand that, and modeling of a lot of data to understand it the best. And then a further small point is to try to also move with this type of data generated with cell cultures into getting estimates of exposure, translating that. We're working, I'm saying a lot of work, which is not finished and perfect, but ongoing, and it's exciting me as well. And that's why it's more fun to talk about it. And finally, of course, this is gives you that first tier overview, and you can move to the second step. So that's what I'm getting you the idea. So what do we do here? We have 5 assays in this 5-score screening, which are done on the high throughput cell number changes, DNA damage, cell viability, ATP content, really typically oxidative stress and apoptosis. Only one assay. Usually you should have many assays, but the idea here is not to be perfect in academics. It's to get an idea. The first tier to then move to deeper mode of action analysis. And then we do the multiple points of departure. We do it by benchmark dosing, a lot of different things. So I'll show you, I'm starting to show data in just a moment, because this, of course, is primarily conceptual for most of you, but a little bit of data should be there, but not looked in depth. It's to give you an idea how we move primarily. So we have ranking. We have that from certain days, we're lowers, we have from benchmark modeling, you do active concentration 50 or inhibitory concentration 50. Very classic. You have this score of ranking. You have Tukey trend test, which I just show in a moment. We have grouping. See, grouping is a cool thing from the governmental point of side in making persons interested in toxicology. If you can, it's also called read across when you do it more in a more larger sense, but then it's more tricky. But grouping, do certain substances from pattern, analyzing the pattern of toxicities, can we say that they are very similar? These kind of things. Here, in this case, not looking at primarily structure, but looking at biology. Okay, so that's pretty much what we do. So here is just a little bit. I'll get into data in just a moment. Here again are the 5 assays. We do it over time, also to longer time, and it's with materials. It's proven to be actually really important, because many effects are delayed. They leak ions and so on, and break down and leach under the culture condition, and the toxicity may only be apparent at late times. And essentially what it is, you look at area under the curve, you analyze that, then we use this kind of tool primarily for the scaling and normalizing it. You can also do this kind of cake diagram, so one assay could be a little bit more than the other. And then you merge that essentially into a visual one. And with this scaling, you make it from sort of 0 to 1, and that gets you into a heat map, and you can rank. And essentially here, it's a scoring and ranking process. You combine all the assays, you get a one endpoint time point concept with a move to a sort of multi-time endpoint concept. And we cover many things I really mentioned already. You have to always consider those interference issues, and you scale them, and you have an output score to apply to ranking, hazard severity estimates, and grouping. All right, so what are we studying in there? Harmless case studies. We're studying from paper making, from paint. These are silicas primarily. You have catalysts, which are there, and then you have also insulation. And you also have agriculture, and particularly we found, which I'll show a little bit more maybe data on, or point out, that perovskites, which is one group that we're studying, and immogolites, which is another group, are actually really toxic when we do this first round of analysis. All right, probably the most boring slide in the whole presentation, but you got to show you at least something. A lot of different nanomaterials for which there is a lot of information accumulated already. Carbon nanotube, multi-wall, single wall, graphene oxide. These have been huge projects in Europe. Lots of money in application. Carbon particles, clay, silica, mixed metal oxides are very often standard reference material with copper oxide, and also titanium oxides are there. I won't go super deep in these things, but rather doing the biology for. Okay, finally some data. So here we can look at the 72 materials, and the Tukey trend test is very good because it combines it all. And for those who are really interested in details, you can read below. But essentially what you see, you move from, these are the 4 cell lines, right? And you can also see the 5 assays, right? Endpoints. And you can see that, of course, brown increases with time. It increases, these are the case study materials here. And you can see they get really brown quickly. You can also see that the THP1, the macrophage line, is maybe a little bit more brown than the other ones. You can probably also spot other cell type differences, but you can realize that some of them are quite toxic by these kind of analysis, and some are very inert. Okay, then you can also look at the different loads. Of course, this is all by informatics. Nobody can just sit. You have to have code writing for every little analysis. It's just informatics. And I didn't mention earlier the sort of AI aspect, but I'll come back to that. I realized I sort of didn't go deep in there. But here we have really informatics. And you can, of course, see that it can vary. It can vary between cell lines. You see the 5 assays. You see them sort of ranked a little bit from activity in this kind of way. Here we can maybe stay a little bit longer. You can see different patterns from the carbon nanotubes and the single wall carbon nanotubes. And you can see some are actually like very inert, some of these clays. You can also see that the perovskites come very strongly at the 72-hour time point in actually all of the assays. So you can actually group them, which I'll show in a later slide, very much by this type of analysis going very broadly here. You can see that certain materials are more active in one assay than the other, but there's also interference issues here. So part of that you have to really be careful when you analyze. And you can also see here, for example, that the silicas are very active on the ATP level in this A5499. And when I go over and people say, oh, do you really believe this? Is this true? Is it really there? And they repeated it all over this last summer, and I've accepted this is true. So cell line differences, not that you totally understand them always, but very often you see, as you saw with the Tukey trend test, that the macrophage line has more. But you see patterns, and you can use the patterns. Now, a very important thing with materials, again, everything is done by informatics here. Nobody can sit and do this, and there's no manual. You feed and you write program. Everything on the material side is also not only mass, but it's surface area. So you can see that certain materials are here on the surface area. The toxicity is actually considered bigger when you do all these rankings of them. And see, other ones are they're black, they're just straight, and other ones go down. We can come back to this, but it's just to give you ideas on how you need to work with materials. Here are a lot of ranking methods. So we go all the way from the TOX5 score, that slide that I had, where all the analysis and the point of departure curve, if you remember that slide. So here they are all together, and it's by heat map, and you can see that these perovskites come up they're very toxic compared to many other ones, and some aerogels for insulation, for example, are quite inert. I already said that. And you can see kind of patterns in this way. So you can rank them, and here they're ranked together, all of them, but they're just grouped so that you can see the reference materials versus the case study materials. And here we're back on that slide that I spent a little bit more time on. But the key point here is not look at here, but look at this end, because this is grouping analysis under doing benchmark dosing. So we have going on the curve of the dose response curve and looking at a lot of different data points at different levels of toxicity and see how it influences the grouping. And essentially here, this is not heat map. The color at the end is just indicating another group. And except for some of the silicas where you see a mixture of colors here, they're all grouped separately. And that's quite nice, actually, by these assays. And here's just an example on that, showing that these perovskites, which had a very particular, now we are analyzing 60 parameters, right? 4 cell lines, 3 time points, and 5 assays. So that's underlying in the informatics analysis. And we can see that on different levels on the scale of the point of departure assessment, we can see variable groupings, as you can see. And often, or sometimes there are also other materials that come up. It's just examples for you of the type of data that you generate with this tiered approach idea. You can also use hierarchical clustering, actually usually defines into further groups very often. And here is the TOX5 score alone. So you have these parameters, you can see 2 larger groups, and then you can have smaller group based on 95% replicability. And there also, I can't see from here, but I know there's like a 98 or something. So you would even have a smaller group. Actually, you have you see 12 here on 2 or sort of 14, but it's actually 21. But the point to make is that we can already, by the high throughput analysis, and then distinguish these and separate them in groups. All right, let's get a little bit mode of action thing. What is kind of cool here? Then when we have the ATP analysis, we have the DNA, which is sort of reflects cell number. We have also the reactive oxygen species, because that's really important for inflammation and toxicity. And we have DNA damage repair. And we can sort of look over here. You can see that the Bs2B cell line, the immortalized one, it comes quite high on these kind of things. We detect a lot of toxicity there. And we also, with the THP1 cell line, detects effects. And here we have caspase. So that reflects apoptosis. And if I would judge here, because I do, how do we move forward and into the omics? So that is ongoing work. And then we will be using the Bs2B and the THP1 cell line. They have different protocols. We can't use exactly the same protocol for the omics, which was my dream. That was my Nobel Prize dream that didn't work out. We lost a lot of time on Roland's dream. But anyway, we tried it. And so forth, and so we know how to move forward. But it's just giving you ideas of these large big data, looking at many things simultaneously, and how we move along when we do these kind of things. Finally, here's an inspiration for us from a paper that many of you, I think, know of, the Friedman paper. And it's a slide that I think, I don't know, Thomas shows it, but at least your colleague Nicole Kleinstruer, or a collaborator, shows that. And what I would like to do with in this project, and I'm really the one who drives big data AI in this project. I'm very alone, I can tell you, but it's a challenge. But anyway, is to try to do what they've done here. So they have point of departure with the new approach methodology. And what they find is that these provide conservative risk estimates for chemicals. Now, I want us to look at, is it the same for materials? And it is because we do have from bronchiolar lavage, BAL fluid, information from most of these materials, some of the case studies, but all the reference materials. So we are going to try to analyze, and it's ongoing. And Pekka is doing wonderful benchmark dose analysis just recently, also with the traditional, which is there, the animal experiments on the BAL fluid. And we're trying to see, can we see something similar from these material studies? So a little bit on the data. Where do you get when you start with the materials coming out of that? We're about 150,000 or so data points coming out of the assays, the high throughput screening assays. And then you can see you generate like 3 times more data, landing in 500,000 data points. And, it's quite good. It's quite good if you think about how we used to do science. I'm an old guy, as you understand, and getting 20, 30 data points from a big cell culture experiment. It was 1 or 2 weeks of work and not too far time back. So you can see what the different points of departure, the ranking, the various scoring, the lowest, what they do. And I always encourage all the people that work with me and sometimes listen to me, that you have to show the data points. You have to know what is data, what is generated data, where does the data come from, what is sort of raw data, and more in AI, as AI can't discriminate. This is a big issue, actually. So I always say you have to show your data points. All right, concluding on the first part, and I will move faster on the second part than I had planned to. So the high-throughput screening permits taking a data-driven approach to generate hazard severity, estimate spectra, point of departure, very important thing. Go to ranking and to grouping, and from getting regulatory agencies interested in the NAMs, if you can do grouping, right? Then you have an issue, then they are listening. And see, the next part is to anchor it further, because they do like the adverse outcome pathway framework. That is that you can take mode of action analysis and understand that to a level. And I'll show that really briefly in a second. Okay, and then you have all of this safe by design, very important, I would think, for an engineering, environmental health and engineering department. This could be interesting. Safe by design, your consideration, whether it's drugs, chemicals, or materials, where you have this possibility for comparison to multiple. It's very good. All under the idea that you can have a testable avoidance of diverse toxicities, of course. Now, then we also have this other thing, which I just touched upon here with the Friedman study, the point of departure focus analysis of the permits or potentially translating these new approach methodology into risk estimate and good weight of evidence are useful to these kind of things, decision support system frameworks, approaches to testing and analysis, etc. And also to say here, this is actually the point. This is the moving from in vitro to translating to sort of milligram per body weight, etc., etc. And I'm not an expert on that, but I'm learning every day. I'm learning something new when I have all this fun with science. Okay, and then, of course, is to inform the next tier. That was the first very slide with the enormous amount of information where I don't know if you remember everything. But the idea first looking at many concentrations and then moving on, so a deeper one. And I actually think that this effort in the materials field even is a bit impressive also in the United States. Because I think it's the largest source of comparative materials in vitro toxicity data. And I'm glad hopefully we have persons interested in studying this and using it. All right, I'm going to do another just maybe 15 minutes, sneak a little bit over my 45 minutes. I hope to keep attention. So what is the tier 2? We applied this tool, the predictive toxicomics-based tool, that was very much inspired by the discussions 2000 and so forth. And my thoughts were very much on looking at chemical space versus biological space. I had a really good talks this morning with Thomas also on that, and with the idea that it could do something. So we generated a tool and by machine learning, essentially from the connectivity map, for those who know that. I don't have time now to go lots of detail, but just to share with you. And we compressed the connectivity map to 100 components by machine learning. And then we found that 14 of them, by reviewing that to another database with toxicity, were highly coupled to toxicity. I'll show what those 14 components are, the number of genes. And we've created a big tool with informatics access and AI handling, a lot of different data collections coupled to our tool. And what I will just show you a little bit for crystalline silica, I've took a material, the work in HARMLESS is still ongoing. So I've selected a literature study of a published paper, just as an example, to share with you how we are currently moving onward also in the HARMLESS project. So how we do omics-based prediction of toxicity, how we do mode of action-based grouping also on the omics level, and how we map all that to the AOP, the adverse outcome pathway framework. That's maybe a second thing to take along for those who are not into this so much. NAM for new approach methodology, and AOPs. You got to learn about AOPs. Then you have two things with you from this lecture. And we've generated these tools some years ago, started actually around 2009 actively, and published on it when I was finally thinking that it was a useful tool, 2017. So what are these 14 components that we have here? Well, they are here. They cover many, many things. And you'll see in just a moment that crystalline silica is activating pretty much this whole group from a publication that we're taking. But you can also see that we have cell cycle coverage, other types of tox mechanisms covered here by this tool. Oops. So we've created something that I termed then the PDGS AOP OM. And the idea with this whole idea with the AOPs, and this is from a paper that we've adapted it here, is that you measure all these early things on a molecular, organelle, and primarily cell culture level. And by looking and finding key events, molecular initiating events and key events, you're able to predict on a tissue, organ, and organism level. So that's the idea. You measure and you predict. And we have generated via the gene ontology, which is sort of a language in simple words, how you can analyze it. And I'm just showing here the lung AOP. We can handle 28 AOPs, which are important. They are coupled to inflammation, cancer, fibrosis. I'm just show fibrosis is because I'm going to talk about that as an example. A lot of people in toxicology are interested in fibrosis. I'm equally actually interested in the others, but you have to choose something. And you actually, here are the components of that where you just saw. You can see that that cluster that I just indicated, I don't know how to go back, it doesn't matter, is covering there. But you can also see that the predictive toxiconomic space components cover these. There are another 12 or so AOPs in there, but they don't have gene ontologies in their description. So then we can't analyze them by omics. So it's going over the gene ontology description where we use it. Okay, so here's a study on crystalline silica, and it will just prove as an example for you on how we do. It has a very acute inflammation, large surface area. We talk about hundreds of square meters of surface area for just a gram or milligram. So I mean, for many types of nano. So it's really there. It's inflammatory. We have reactive oxygen species. It's insoluble, so it's retained in the airways. We know silicosis, very strong induction, which is a form of fibrosis, and if it's left, also has moves on to cancer. So we know that already. So let's look at the omics data from that paper in a moment. So here you can see that now we have it's conceptual. Just have to sort of hear overall. What do we do? Well, we look at we get a score for the predicted toxicity space. It has to be significantly different for the control, and versus a reference data set, which is the connectivity map, we have to pass over a threshold. And by passing over the threshold, we get the lowest observable signal for the predictive toxicity space. In this case, the lowest concentration tested was actually quite toxic, and we get a very strong signal. We then analyze the components, and then we can see also that the lower has a whole range of these components, which we couple with a cluster of toxicity. Then we move into analyzing APs, and it's based on component correlation. If you do it just with gene expression profiling, the AOP descriptions are so similar, you just land in chaos. You don't get any specificity at all. We worked a long time to get specificity. And the way we do that is looking at correlation analysis of the components. And we also do it by decreasing the gene ontology size. if you think about, I'm a mode of action person, and someone describes an adverse outcome pathway and says, oh, it's increased gene expression. Yes. And for me, who are looking for specificity, that's not particularly exciting. You don't want to talk negative to anybody or be negative, but the point is you want to have specificity. So what you do, you accept smaller and smaller sizes. You just cut smaller on the ontology and get higher specificity for the mode of action analysis. And that's what's shown here. So the algorithm developed to be detecting as much as possible, have positive signal for all the 28 AOPs in that first lowest analysis. So that's what that shows. Now, then you can do a phenotype analysis. You can annotate all those 28 AOPs. And what you can see is that quite a few of them are getting signal, and that includes cancer, fibrosis, and inflammation, and also at the low one. So a reverse translation in a programmatic analysis of AOPs under the stringency of the Lowell analysis corresponds to known effects annotation. We have inflammation, we have many AOPs for that at the A2000, so that's sort of in intermediate, decreasing the size of the ontologies that you accept for interpretation, and for cancer and for fibrosis. And let me just show you fibrosis. I'll go another 5 minutes about, and here it is. So here we are. This is the fibrosis AOP. It's actually a OECD endorsed, so it has a new number 33, but otherwise in the wiki, AOP wiki, which is a living document of descriptions of these AOPs, it has number 173. So here, what can PDGS do? Here's the MIE, molecular initiating event. Here are the key events leading to the adverse outcome in the end. And so these three are covered by the predictive toxicity space. And here you can see the lower one has the ABC. It has also C here for this event, which is covered here. And it has also these events. So you get actually a qualitative and quantitative output to each of these 28 AOPs when you do this type of analysis. And here's an example of visualizing this with eye diagram. This is just showing, this is comparing to another 20 materials in 6 other publications. But it's just an example and to share with you. And over here is a sort of explainable AI concept of showing how the components relate to the event level and how that relates to the AOP level. And here simply, we just notice this one, we notice this one, which are there, and the 3 events which are captured here. So it's just a way to show that such a data for you. I realize I'm losing some of you, but maybe you still get a sense for what we're doing here. So how can you then use this? I showed you also grouping from the high throughput data, and now let's do grouping on the omics level. So we can do it on the component basis. So that can verify the mode of action driven toxicities. You can group on that. In this case, you can see that this cluster 2 here had a lot of these components active, and including this compound Minusil, which also DQ12 is a term that we use for it sometime. You can group them. So very quickly, you can also group them with high specificity on the, it's all component correlation based, but you can group by the events activated. You can get output programmatic. All of this is programmatic driven analysis for each of those AOPs. I just showed you one. And you can see that the predictive toxin space is very active in different clusters. And then you can also do it on the AOP level. And DQ12 there means essentially this one. So that is what you can do. So let me just close with a few couple of slides. Hopefully not overdoing it too much. This predictive toxicity space was constructed to decrease uncertainties in results. It's built on big data and explainable AI informatics methodology to the degree it can. It's absolutely not at all complex in general terms when you do the analysis, but a 14 component algorithm. We are very good to do flat 2, 3 space, but 14 Can we think about that? Very difficult. Maybe some can, an extra dimension of 4. I think I stopped myself at 3, but here is working in 14 dimensions. Now we can predict cell and organ toxicity on a very transparent 2-component scoring metrics from applying that algorithm. This has to do with getting statistics, a gene set enrichment analysis, and it also referring to a reference data set. And the idea, of course, is that effective and validated toxicity effects prediction implicates applicability very broadly to safety assessment. It's not only for materials, but also to consumer product generally. And we provide a toxicity hazard warning with that. And the idea, this I realized went really fast, is that we found it further, and we have all the regulators in mind. Regulators like grouping, that then we can maybe save efforts. We can group things together by toxic mode of action or other types of mode of action. And here we are also founding it further by informatics analysis into the adverse outcome pathway framework. And of course, this is broadly applicable in many different ways. And just to say, are we doing something totally unique? No, Thomas already mentioned, we are all listening to one another. And this thing came 2019. And if you look at that from the EPA here, there we have high throughput, we have transcriptomics. I think you would first have an idea about toxicity, which, you know, I would be better. You can't do millions of, it still is a bit of cost. When you have no defined pathway, you sort of walk down. Where do you land? Point of departure, low well versus no well, etc. Going up and down the point of departure curve like that. Okay. Now, if you have a defined target, well, here we are where you are specialists. Cooler analyses like microbrains and all of this. I'm showing simple cell lines. That's what we know how to do. We get an idea with that. But can you move to a better level and use other ones which are even more predictive, optimize organ? But you can still see with AOPs, existing AOPs, okay, then we have estimate point of departure based on AOP. I showed you such data. I tried to show you, at least I realized it fast. Or you have point of departure when you don't know the AOP, without the AOP. But you can see, are we being inspired by other people's thinking? Yes, of course we are. So finally, I think this slide, and there's also references, general slide. And for all of you in the department, please, you can get a PDF of this one if you like. But I've had this slide. These are a lot of projects. You know, normally you have to do these project slides, and it takes half of your lecture almost if you work for so many years as I've done. But here you can see the high content information. This is sort of high throughput approach. And of course, you come in with structural alerts, as I showed, and then you have dose response. You do that on the high throughput and by the scoring concept. You have computational models, and you're looking for relevant toxicity pathway. Then you may have kinetics. I didn't talk about that. We've done a lot of work in other contexts of drug-induced liver injury on that, but that was not part of this one. But then a risk assessment based on that. And now with this little dimension coupled to the Friedman study, that maybe we can also translate some of this data to the type of things done normally with exposure assessments, these sort of point of departure from NAMs versus traditional or exposome. You know, there were many things that I didn't have time to talk about, but that's what it is there. So there finally are some slides for or papers which tells a little bit about these ideas. Thank you so much.
This is down there, so I can turn on the microphone.
Okay, good.
You can call on people that raise their hands.
I think my good friend Ed is nice.
Well,
He's probably planned a question long time. Yes, please, Ed.
Thanks for your very provocative thought and the amazing work you've done. And of course, you might not call me a friend after I ask a question here. Is your 4-step human cell line panel really adequate to assess all of these effects? And how can you validate that? And I immediately think of, you have one monocytic leukemia cell line to represent the immune system. And the immune system not only has macrophages, monocytes, but a whole lot of other types of cells.
Yeah.
And then even looking beyond human cells, what about the microbiome, which we're increasingly aware affects a lot of human diseases.
Absolutely.
And you could test a panel of cell lines and it looks like a harmless chemical or harmless particles, and it could affect the microbiome in a way that can lead to human disease.
Yes.
And where do you decide when to cut off the panel, how do you assess whether or not that's robust enough?
Yeah. I'm ready to come back with an answer, but if you give more questions, I will forget the first part. but anyway, if I take the microbiome, yes, of course, you can't be an expert on everything. And I think it's a very valid point. And in some discussions we've had here, maybe we are not at the tipping point where all of these things are already there. They can be used to replace, more traditional testing. But you can't study all. Now, these cell lines, why are they there? You missed actually something that I may try to make a big point of, as I see it. And as you're a really good friend, I can be really direct with that. The first part is there to give an idea. It's not to do with the traditional science. It's to set a stage where we have an idea. And cells are cells, and you can argue about it. The idea is with 4 cell lines is to use at least a bunch of them where you have where you can compare them. The other important point, of course, I didn't mention is that what type of cell lines have been used so far in nanomaterial studies and work. Those are those cell lines. We have a reference to everything. We can get people to be interested in our work. If we take a new model that they never studied before, they are studied from ease to use and to know. So this is what it is. But of course, you're also bringing up this idea where your department here are word expert and making the mini brains, for example. So you combine many cell lines together. We've been part of several projects where we try to do that with variable success, where we combine different cells to get a better concept. So we're aware of the point you're bringing up. Here, the idea was, let's get a first idea. Let's use cell lines that people have been doing. Let's get a first thing and then move and deepen the acceptance by proving at the AOP level if it's quite correct. So that's the idea. Love your question. Yes, you back?
Hi. So I'm interested in, you showed that you're looking at like the cells and it's predicting for tissue and organ and then ultimately going to individual community. Is that like the planned step? Like that's like the goal. So like how do you plan on validating that?
When the sort of it's good. You're giving super questions already. Big data first of all. There's a lot of data there. We need comparative data. That's one part of it. And if I say that we probably generate so far globally the largest effort of big data on materials and sharing with you, that's one step. And having it under, in a lab under a specific thing where you really can compare all this cell line. Now, a second thing is I still wanted to share with you that already on that initial level, we actually can group the substances. We can see from the patterns that the different types of reference materials and the case that I didn't go in deep in that, but because I wanted to give you conceptual lecture, we can already group them, to my surprise. Of course, how would I know? But with this large effort, we can group them. Now, the AUP thing, see, that's so hard to communicate here. This is where it comes, how do I get the regulators on board? They sort of know less than I think all of you in the room do, and we want to get them on board. They get on board if we can show that we have groups. Then that makes them interested, because then NAM technology, new approach, it's not new. If you heard Thomas' lectures, he said it's not new. It's just a name of in vitro, in silico, etc. But then they are interested. And this next thing that can make them excited is you anchor in the AOP framework. All of these hundreds of scientists that work together to describe what are the key things that must happen before fibrosis, a cancer, an inflammatory, all things. They all work separately. Describe. If we can do that, then we anchor. See, then we get them also involved. So, all of this goes very quickly, but your question makes me able to say that again. That was the idea, to show that. But of course, there are a lot of surprise findings when you do science. And information technologies is absolutely key here. You can't do any of this without writing code. It's code, and it takes forever. Believe me, when you're waiting there, I was trying to support and lead people, which I at least used to do. Now they're my collaborators, so they lead me equally much. Thank you for that question. Yeah.
Hi, Venice Lier. I had a question about the exposure route in a sense, because this is something that in vitro assays tackle it or have to tackle. For example, these nanoparticles, I looked up some of them that you mentioned, and some of them might be, for example, a part of the solar cell. But
yes,
how does the route of
perfect.
exposure get incorporated into this framework, into this pipeline? Because adding something to a dish, a cell line, is something, putting something in a liquid culture, but it might not be the way how people are supposed to. How did you tackle that? Were you able to tackle that in your pipeline, or and it might differ, right? The same as with the discussions that I had with my friend at the FDA that tried to regulate the vaping. All these chemicals that have never been vaped and inhaled before are now
exactly.
being inhaled.
Yes.
Route of exposure matters.
Yes, absolutely. Yes. Yeah. Your question sort of relates to this major thing where I try to tell you I'm a small part of the puzzle. And the first part is the problem formulation. This thing, are they really exposed? Do we do it? Do we, from the general, you really are saying the hotspots of concern, where do we want to go? Maybe we can't test yet everything. Maybe we can almost test everything if we adapt these, although the cell models are lousy, etc., etc. I'm not fond of these cell models, but we got to do something, and we need big data. And then we go see then what do we know? And then what we don't know, then we of course make data, we're quizzing data. And this is, I want to now almost walk over here. I'm not allowed to, I guess. I'll just do it anyway. Okay, so where am I? I'm on this one. Now, what they do, right? The human environment toxicity, is that's where I'm. That's the little part of the puzzle that this lecture is about. I stand for something, and I'm part of a super group of people, the scientific community, which is in materials and is doing that. So that's what I'm doing. Of course, I depend on them. What they tell me is important to analyze. I provide them with knowledge and, on cell biology, and this whole idea, what is the main thing in terms, so that we get the regulators to like this. So I keep saying the same thing. Maybe I'm not answering nicely, I'm just babbling now. But anyway, I'm excited. But you see where we get the grouping and the anchoring in things that they believe in. We can't get to them. This is why we had a policy. We talked about policy and all these type of things. I'm sorry, there was someone there, and then you want to go.
This is related to her question.
Okay.
You talked about BALs. Remember that? Yeah, So, it's like when they put saline down into the rockets and they pull it back up. So it gets all the cells that are there and any particles. So you're going to do something with that, right? Are you going to incorporate that into your tiered approach? BALs?
Oh yes, the BAL is not me. I'm doing this, but I'm collaborating. I'm not an animal. Sorry, Kathy, you know that. And it's not like, but it's just like my specialty. So that's the Friedman idea that is have here, and that I think is cool. We can do that. Sorry, it's coming soon. Oh no. Okay, so it was that sort of. I just see them. That's sorry. it's a point of departure, and it's with the dose. And the idea of that paper is that 90% of the POD measurements with new approach methodologies are conservative, and which is better than if they were not. They're on the side that we overestimate risk. Of course, we want to have better precision, the best precision we can exact. So we are going to test comparing to valve fluid, bronchial lavage fluid data on neutrophils, eosinophils, epithelial cells, macro, I mean, they're all kinds of super data. So that will be a cool, being inspired by this art. I wish I could take the screen and show you that. I really can't. Yes. Yes, Bruce has been there. Yeah.
I'm kind of curious on how these kinds of models are actually used in the path towards commercialization of compounds.
So, sorry, towards saying.
How these kinds of models are used in the path towards commercialization of compounds. So if I have a handful of compounds that I'm interested to commercialize, can I send you the names of those compounds and you'll give me some measure of predictive risk, and what would that measure be?
That's again the big scheme. You get scores, you know, in this way you do get scores. I like the last part of your question because it's really challenging me to see. I think you can, first of all, I say you would probably do it primarily blinded, because you know you never want to know. You want to just get it. You first do the work. You don't want to tell the. So usually that could be a blinded thing, at least at the beginning of a corporation, but then afterwards, when you've done many rounds of a commercial testing, you maybe you can say, we believe this is toxic because you trust each other. But otherwise you actually you get you can get a composite score, and this is where the IATAs come in, how they are used, whether they are intelligent. The I think others should say, but at least it's an ATA, let's put it that way, and then others can say if it's intelligent, but it's a term. So what you do, you go down, do they have these properties, do they leak ions? Okay, worrying. Okay, we can't say. Do they have a very long asbestos-like shape? Yeah. Okay. Problem. You can't just go. I mean, and so you have this kind of tick of box system, and this is also done. You can go to the Harmless webpage. I just shared today my little piece of this. So you have decision support systems, you have tools where you go in, you ask your questions, and they're public. And if you talk also to Thomas, he's often telling about to have this public, everybody can go in, they have their molecule, and they can get an idea about how toxic it is, and that there should be public such tools. And this is where good use of AI can come in. You can't totally trust it, but you know, it gives you hints. And by the way, I tell everybody, use ChatGPT as much as you ever can. Don't believe in it, but get it as a co-pilot, as a partner. Talk to it, use it. To me, I'm totally open. And I stand in these projects with traditional scientists in Europe. I can tell you, I'm the only one who says that. So I'm more in your lines, although I'm far away from here. Anyway. Well, wonderful. A fantastic number of questions. Yes, please, of course.
Thank you for the great talk. I'm really interested in how you can integrate the toxicogenomic data sets into 3D stem cell models. Specifically, I want to know what advancements or methods you think are the most vital for regulatory frameworks that can translate to real humans.
When you do all these other things, see here, I think with these old-fashioned thing. Also, we think about material. If I just bring it back to why did we use these silly cell lines, you know, tumor-derived, really strange, not differentiated, and so forth. But that is the materials, but how do they penetrate these kind of things? You know, how are they taken up? How much of the material effect is on the surface? How much is because they're taking up? I'm not even showing that to you.
Yeah.
I'm out there in the blue. I don't know that. You see, but we got to start somewhere. We need big data sets. So If you can do these 3D types of culture, are there more complex ones? There's lots of fantastic work on that. I've, you know, over the years, I had groups of people doing that. I was never really happy in my context, whether this had to do with the persons, whether or with the reproducibility. If you have that, I think you have people here that you can put this question much better than to me. But we have very much tried it, and we've been in a project called Patrols, for example. And we did that. We did small liver spheroids, and it was a 4-year effort, and I wasn't happy. It wasn't reproducible enough. I'm sorry. There's a lot of this kind. You have to try, but you know, then in the end, I'm such way. I don't want to go with it. I share it like this. I talk about my absence of knowledge and everything, and how I try to find it. But going to the full way of yes, this is good. You know, it takes a lot for me. Thank you for the question.
Thank you.